Hello everyone again. So we will continue apoptosis. Then next we will study about the extrinsic pathway of apoptosis. So in extrinsic pathway of apoptosis, the death stimuli or the death signal is not generated within that cell, but it comes from another signaling cell. Okay, so the death signal comes from different cell, and the cell which produces that death signal is called signaling cell. Okay, and signal comes as a ligand. Okay, ligand. So external signal ligand comes and binds to the receptor molecule present on the plasma membrane, and once the death signal attaches to the death receptor present on the plasma membrane, this pathway this pathway will be activated, and that is called extrinsic pathway. And since you can see so many caspases are involved in this pathway is also known as caspase dependent extrinsic pathway. Clear. So intrinsic pathway was called mitochondrial mediated pathway. In this pathway, so many caspases are involved, and that's why this pathway is also known as caspase dependent extrinsic pathway. And also a receptor is involved, death receptor is involved. So another name of extrinsic pathway is death receptor mediated pathway. So it will have two names: caspase dependent extrinsic pathway or death receptor mediated pathway. Clear. Now let's see about the signal, death signal or the ligands. Okay, so in this picture, you can see the death signal. It is written TNF alpha 1, that is tumor necrosis factor alpha 1. Then there is another ligand also, it is known as FAS. Okay, FAS. So the FAS is actually not shown in the picture. In this picture, only TNF alpha death receptor is shown, but there is another ligand and another receptor also present, that is FAS, FAS ligand and FAS receptor. And the name of the both receptor is actually the death receptor. Okay, and this one binds with TNF alpha. That's why you can see it is written TNF alpha receptor. But there is a another ligand called FAS, and FAS death receptor is also present. But unfortunately, I do not have any picture of it. So the pathway is same, fully same, but that receptor picture is not with me. So let's understand about the ligand. So the ligand is either called TNF alpha, tumor necrosis factor alpha, or FAS, FAS, FAS. Okay. So you can see both are trimeric protein. If you see the structure of the ligand or the signal, they are trimeric. Okay, they are trimeric. That means it is having three subunit. And TNF ligand is produced by the macrophages. Okay, so the macrophages, they are helping in our cell immunity. And another one is called FAS ligand. So FAS ligand is also a protein, and it is produced by natural killer cells and T lymphocytes. So both the ligand, okay, both the ligand TNF alpha and FAS are actually produced by those cells which gives us immunity or which protects our body from diseases. That's why this type of extrinsic pathway is only activated during virus infection or any pathogen infection. Clear. So this type of apoptosis pathway is usually generated during infection time. Okay. So now let's see about. I have already mentioned the structure of ligand, the signal. The signal is trimeric. Both TNF alpha and FAS are actually trimeric, say having the same structure. They are trimeric on the nature, and if you see, both are present on the surface of the signaling cell. So the cell which produces, you can see it's a signaling cell written, and the signal is actually present on the surface of the cell. Both TNF alpha and FAS are TNF will be present on the cell surface of our macrophages, and FAS ligand will be present on natural killer cell or T lymphocyte cell surface. And TNF alpha binds to TNF alpha death receptor, whereas FAS ligand will bind to FAS death receptor. So the structure of the death receptor is also same. Both TNF alpha death receptor and FAS death receptor is same only. These receptors will have a ligand binding domain. So this one, this ligand binding domain, ligand binding, it's outside the it's facing outside the plasma membrane. So those are exoplasmic domain. So that is, so all both the receptors will have ligand binding exoplasmic domain, then a single transmembrane domain which you can see is embedded in the plasma membrane region, and a cytosolic domain which is the the receptor protein portion which is facing inside the cytosol. It is a cytosolic domain. So both the death receptor will have the same structure, a ligand binding exoplasmic domain, and a transmembrane domain which is embedded in the plasma membrane, and yeah, the cytosolic domain which is facing the cytoplasm. Now let's see how the pathway works. Okay, so the TNF alpha ligand will bind to TNF alpha death receptor. Same, FAS will bind to FAS death receptor. Clear. So FAS and FAS death receptor is not shown. You can just note down. So TNF alpha will bind to TNF alpha death receptor, will bind to fast death receptor. Anyways, whenever the ligand binds to the receptor, so whenever TNF alpha binds to its receptor, TNF alpha death receptor, or whenever FAS binds to fast death receptor, a conformation change will happen in the receptor. Okay, a small conformation change happens in the receptor, and and as a result, what happens is, yeah, the death receptor, uh, the death receptor complex can bind with certain proteins. Okay, so here it is TNF alpha shown in the picture. Okay, so when it binds to the receptor, a conformation change took place, and you can see on the cytosolic portion of the receptor, yeah, some proteins are attached. So that's what happened when the ligand binds to the receptor. Whenever TNF alpha or FAST bind to its death receptor, a small conformation change happens in the receptor, and it leads to some of the complex protein binding on the death receptor. So here you can see two proteins are there. Okay, TRAD, TNA receptor associated death domain, and fast associated death domain protein. So two proteins come and attach to the receptor whenever the ligand binds to the domain. Okay, okay. So whenever TNF alpha is binding to the TNF alpha death receptor, yeah, you can see on the receptor uh, both will come and bind, TRAD and FAD. When FAS will bind to its fast death receptor, yeah, only FAD will come and bind. Okay, TRAD will not come and bind, only FAD will come and bind. In the case of FAS ligand and fast death receptor alpha. But in this picture, you are seeing TNF alpha and TNF alpha death receptor. So yeah, in the case of TNA alpha signaling, TRAD also comes, FAD also comes. But if it is FAD, if it is fast ligand and fast death receptor, only FAD will come and attach to the receptor. Anyways, FAD is common in both, and TRAD will be only coming and binding in the case of TNA alpha, binding with TNA alpha death receptor. Okay, anyways, FAD will be common to both. Okay, so whenever, yeah, ligand binds to its death receptor, TRAD or FAD will come and bind to the receptor. Now this FAD, when it has come and attached to the receptor, it acts as an adapter. Okay, it acts as an adapter, and this adapter can recruit and activate, yeah, initiator caspase 8. Okay, so how it becomes active? It's slightly different. FAD actually helps in dimerization of initiator caspase 8, and it makes it active. So here you can see that once FAD has come to the receptor, it is activating initiator caspase. And you know that initiator caspase, what it does is it activates, it goes and it cleaves the pro domain of all the effector caspase. So here you can see all three are mentioned, caspase 3, 6, 7, all are effector caspase. So this initiator caspase 8 cleaves the pro domain of all the effector caspase, and yeah, it leads to cell death. You know that these caspase 3, 6, and 7 will go and cleave all the cellular cell protein substrates, and it will kill the cell. Clear. So death will happen. Okay. Now there is one more thing. There is one more thing. This initiator caspase 8 activated several effector caspase and it leads to cell death. But this initiator caspase 8 has function, one more function. That is, it goes and it cleaves a BH3 family protein called BID, BH3 interactive uh, domain death protein. So this initiator caspase 8, it not only activates all the effector caspases, but it goes and it cleaves a small portion of a BH3 protein called BID. And that small portion in the blue in the blue, you can see this BH3 family protein, it cleaves half portion of this BH3 family protein BID. And this half portion is called T bid. Okay, small t and bit. Okay. So apart from activating all the effector caspases, this caspase 8 also goes and it cleaves a BH3 family protein called BID, but it cleaves and only the small portion, that small portion is called T bid. And that T bid, yeah, it's a small portion of a bid, and bid is actually a BH3 family protein or BH3 only protein. Yeah, so this T bit small portion is also a BH3 only protein, only it's written also. So this BH3 only protein, yeah, it does the same thing. This BH3 only family protein will go and do the same thing that happens in intrinsic pathway. This BH3 only T bit portion will go and it will bind to anti-apoptotic protein like BCL2 or BCLXL, which will which will allow all the pro-apoptotic protein bags or bad to oligomerize. And once it oligomerizes, cytochrome C easily escapes from mitochondrial intermembrane space. And yeah, cytochrome C will bind with APAF1. APAF1 will activate initiator caspase, uh, initiator caspase 9, and initiator caspase will again activate other effector caspases, and again it leads to cell death. Okay, so in this extrinsic pathway case, whenever a death signal combines with a death receptor, a small conformation change takes place in the receptor, and adapter protein like FAD and FAD binds to the receptor, and FAD acts as a adapter for uh, for activating initiator caspase. So main initiator caspase is initiator caspase 8. So once initiator caspase 8 is activated, it will activate or it will cleave all the pro domains of other effector caspases, and they will be active and they will cause cell death. Apart from that, caspase 8 also cleaves a BS3 only protein called BID, and its small cleaved portion goes and binds with anti-apoptotic protein free and then when this BS3 only protein binds with our anti-apoptotic protein, so pro-apoptotic protein becomes free and they oligomerizes, and yeah, cytochrome C escapes, and yeah, just like the normal intrinsic pathway, cytochrome C will bind with APAF1. APAF1 will activate initiator caspase. Initiator caspase will again then cleave the effector caspase pro domain and make the effector caspase active. Again, effector caspase will lead, they will go and they will cleave all the cell substrates, leading to the cell death. So this is extrinsic pathway. So these are the two apoptosis pathway, intrinsic pathway and extrinsic pathway. So in intrinsic pathway, what happens is, yeah, the death stimuli is generated within the cell itself. If a DNA is damaged, at that time, it can be the death stimuli. If integrin is disrupted, at that time also it is a death signal. Or if growth factor is absent, that is also a death signal. Okay, and the signal is generated in that cell only. But extrinsic pathway means it's a receptor mediated pathway, and for that the signal is produced by different cell called signaling cell, and it is attached on that cell surface only, and it does that signal binds to, yeah, the death receptor which is present on plasma membrane. So it's a receptor mediated pathway, and it is only activated during infection times. Okay. So the signal is not produced in this cell, it is produced by another cell called signaling cell. The signal just comes and binds to the receptor of death receptor. And when it binds to death receptor, yeah, it activates caspases. That's why this is also known as caspase dependent apoptosis pathway. So these are the two apoptosis pathway, intrinsic pathway and extrinsic pathway. Thank you.